This video is protein transport, vesicle tethering, docking, and fusion. So we're picking up with a vesicle which is in the cytoplasm. It does not have a coat on it at this time, and there's a three-step process of tethering, docking, and fusion before this vesicle will then fuse with the target membrane. Some of the things to pay attention to here are that we have a mechanism in place through molecules on the vesicle as well as molecules on the target membrane, which can ensure that the proper vesicle gets to the proper target. And if these things do not meet up properly or match up, then the vesicle would not dock or tether at this location and would bounce off and potentially find another organelle or other membrane. We call these molecular markers, so these molecular markers here are on the outside of the vesicle and target membrane, and they're recognized by one another, in particular through these receptor proteins that are on the target membrane. So let's start with the first step, which is tethering. And just like that sounds like this is kind of the initial tying up and kind of recognition between a vesicle, which is incoming, and this target membrane. So tethering uses a key protein called Rab. And so Rab is shown in yellow right here. Rab is one of the key markers for vesicles to be recognized by a target membrane. And a couple things to know about Rab are that Rab is part of a larger family of proteins. Many of them are different sorts of Rabs, but then they're part of an even larger family than that, in that they are a type of monomeric G protein. These are also known as GTPases, which bind GTP and then can remove a phosphate group to provide energy for some process and then form GDP. You may be familiar with other monomeric G proteins, and many of them have three-letter names that start with R. One of them is RAS, which you would have heard of from receptor tyrosine kinase signaling. Another one is called RAN, which you would have heard of from nuclear import, and Rho, as well as RAC. And Rho and RAC are used in cell migration. All these proteins have similar roles in that they are what we call G proteins. And again, what that means is that this is a protein that can bind GTP, and in particular, it can hydrolyze GTP to provide energy for some other processes. This is a really common theme, the use of monomeric G proteins within different processes, as you can see here for cell signaling, nuclear import, and also for cell migration. And in this case here with Rab, we're talking about how vesicles fuse with a membrane. All those processes are distinct from one another, but yet they share the same common component. And these are each distinct from each other, but they have a common role. Moreover, I think this highlights the importance of GTP in these aspects of cell biology. You hear a lot about ATP as the source of energy, but here we're seeing a theme of using GTP for many different processes. Okay, so back to talking more about the tethering and the role of Rab. So again, the basic deal is that there are several different varieties of Rab on different vesicles, and there are unique combinations of Rab with a particular tethering protein, and this means that a given vesicle would fuse with one organelle versus another. And basically this initial tethering event, Rab, and the tethering protein, that's a key part to make sure there's a good initial fit. Once this fit takes place, then it's possible to go on to the next step, which is docking. Docking then is an additional step of recognition by which transmembrane proteins V-snare, which is shown in red here, a second protein on the vesicle, which is shown as longer in this diagram, is complementary in terms of being able to fit with it to something called a T-snare. And the T-snare is this blue protein, which is on the target. And basically these look kind of like outstretched arms, and so basically when they bump into each other, they can form an association which allows for docking. One thing to notice here is that V stands for vesicle. This is super handy. So the snare protein that's on the vesicle is called V-snare. and then the snare that is on the target membrane is called T-snare. So that's this docking step here, where these two snares come in contact with each other and form kind of an initial closer proximity. Then the third step that takes place is fusion, in which V snare and T snare wrap around each other much more tightly, and they, through this process, will displace water, and they force membrane lipids on the vesicle and on the target membrane into a continuous fusion, and this reorients the membrane lipids on the vesicle to make a continuous membrane with the target membrane. And what this means is that the cargo protein, which had been inside the vesicle, now is going to be released into the now continuous cytoplasm. This all is described in figure 15-21. The next thing I want to talk to you about is what Botox has to do with snares. And so this probably initially sounds pretty strange. Botox, which you may have heard of from everyday life, is something that can be injected into people's bodies, in particular to minimize the appearance of wrinkles. It can also be used to treat migraines, which is why it was actually initially developed. And what Botox does, this is a protein, in particular it's a toxin from a bacterium, Clostridium botulinum, and you don't need to know the name of it, but it's a specific species of bacteria. It makes this very toxic protein, Botox, and this can interfere with muscle contraction. And basically, this can be injected into particular locations, and then for a limited amount of time, say a month or so, depending on the dosing, then that particular muscle would not be able to contract. And again, this has a variety of different implications, but how Botox works is really interesting with respect to snares. So basically, what the deal is that Botox is this protease that's made from a bacterium, and what it does is it cleaves a snare protein, which is required for neurotransmitter vesicles to fuse with the plasma membrane of nerve cells. So basically, Botox is going to interfere with a snare protein, and it's going to prevent this vesicle fusion. Turns out when you Google Botox, you can find a lot of interesting pictures. I thought this one was just kind of interesting because it highlighted a variety of different locations that people might select if they want to consider minimizing wrinkles or lines on their face, and it really is just kind of arbitrary and kind of a big question. So how much Botox do I need? Hard to say. But again, this really does have some important implications for people who have some disorders where they cannot relax certain muscles of their body. Migraines in particular are one of those. And so Botox can be used therapeutically.